Cancer research has made huge strides within the past few decades, from determining the function of oncogenes in the 80s, to finding the genetic predisposition for cancer in the 90s, to utilizing targeted cancer therapies currently. Two cancers in particular that have experienced the fruit of these scientific advancements are breast cancer and prostate cancer, which you can see by looking at their overall 5-year survival rate. And what that is basically the percent of people who have been able to survive for 5 years after receiving their diagnosis. Now, the overall 5-year survival rate for breast cancer is 90%, and the overall 5-year survival rate for prostate cancer is 99%. Those are pretty high. If we look at a cancer that has not experienced this success, we see drastically different results. One example of this is ovarian cancer, which has an overall 5-year survival rate of 47%, making it the deadliest gynecologic malignancy of the female reproductive system. So what makes ovarian cancer so deadly? And part of it is because of how rare it is, but perhaps an even bigger contributor to the large mortality rate is that despite good initial responses to chemotherapy, recurrence is common and often fatal. Because of this, it has become vital that ovarian cancer research focuses on both identifying better biomarkers for disease progression and also better treatments for intermediate and late-stage disease. My thesis focuses on the latter. However, in order to understand my thesis, it might be helpful to get some insight into how ovarian cancer works. So let's orient ourselves to the abdominal pelvic cavity, which is actually down here, but for our purposes, I'm going to bring it up here. Now, as the name suggests, this cavity contains both the abdomen and the pelvis, which also includes the reproductive structures such as the ovaries in females. Now, ovarian cancer, like all cancers, begins when the body cells begin to grow uncontrollably. And in this case, the cells are typically from the outer surface of the ovary and form what are called epithelial tumors. And these are also the most common type of ovarian tumor. The cancer was originally thought to originate in the ovaries. However, more recent research suggests that it might actually originate in the fallopian tubes. So now that we have the basic knowledge of what ovarian cancer is, let's look at how it's characterized in a clinical setting. And this is done in primarily 2 ways. First, clinicians typically look at the grade, so the morphology of the individual cells. How normal do they look? The more normal cells look, the better prognosis they have. Staging is actually quite complex, but for our purposes here, I'm going to give you a very brief overview. So in early stages, the cancer is typically restricted to the ovaries or the fallopian tubes. It could be on one or both. As it progresses into surrounding organs in the pelvic cavity. And then in stage 4, it has metastasized to distant sites in the body, such as the lungs, the liver, and the spleen, and also lymph nodes in those regions. My focus in particular lies in the later stages, which is when the cancer is spreading throughout the tissue. And there's a really interesting mechanism as to how this works. Ovarian cancer utilizes something called spheroids, which are basically these aggregates of detached tumor cells, which are able to travel now in this compact structure in a fluid called ascites, which is generated by the cancer. And using this fluid, they're able to reach an organ, attach to it, disaggregate, and then invade the organ, spreading the cancer there. Now, recently it's been found that these spheroids are typically more resistant to chemotherapy, and one possible reason for this was explored by my lab and involved the cell adhesion molecule Nectin-4, which has been shown to be overexpressed in ovarian cancer tumor and is implicated in spheroid formation. So by creating a Nectin-4 knockdown cell line, my lab has been able to describe the role of Nectin-4 in ovarian cancer cell adhesion. The goal of my thesis is to determine the differences in chemosensitivity of these ovarian cancer single cells and spheroids between cells that express Nectin-4 and between cells that lack Nectin-4, so using our knockdown cell line. And I hope to contribute to research that is trying to find the means by which these spheroids are able to evade such robust chemotherapy and hopefully shed some light on some more suitable alternatives or adjustments to the current chemotherapy regimens. And I hope that eventually this will improve the overall 5-year survival rate of ovarian cancer, similar to how breast cancer and prostate cancer research has been successful thus far.